Hello everyone, so I'm here today at the Francis Crick Institute to learn more about the incredible work that Professor Carola Vinuesa is doing with regards to immunology and genomics. Professor Carola, please may you tell us a little bit more about your work and what you do?
Yes, of course. I'm an immunologist. My main interest is autoimmune disease. So these are diseases in which our own immune system, instead of focusing on fighting infections, fight our own tissues and sometimes cause destruction. And these are diseases like lupus, type 1 diabetes, multiple sclerosis. So we are trying to understand how they come about and what can we do to treat them more effectively.
I understand that as part of your research, you look into antibodies, so B cells, T cells, how they collaborate and communicate.
Yes.
Tell us a little bit more about the importance of understanding the behavior of those antibodies.
So antibodies are very important because they can protect us, right? They can bind to viruses and bacteria and neutralize them. But these same antibodies, right, when they deposit in tissues, skin or the kidney, can cause damage, right? And antibodies, you can imagine them as if they were, arrows thrown by an archer. The B cell could be the archer. And the B cells need to know what to attack and what type of antibodies to produce. And there is where the T cells come, and they could be like the commanders. The commanders inspect the enemy and give very precise instruction to the archers, in this case the B cells, about what type of arrows, when to produce these antibodies, where to produce them, right?
Oh, wow. Okay. And what has been one of the most exciting discoveries you've come across researching more into the behavior of antibodies and immunology?
Yeah, well, I think the most exciting one for me in the recent years, but also because of its impact, is we've discovered what we believe could be the central pathway or the central cause of the autoimmune disease lupus. We initially did some sequencing of the genomes of children with a very severe form of the disease. And we found that some children, only a small proportion of children, have rare gene variants in a gene that was then not known to cause human lupus. This is a gene that encodes a receptor for RNA, small RNAs. Could be viral RNA, but could also be our own RNAs. And what we discovered is that when this receptor is overactive, it makes rogue B cells make autoantibodies. And initially we thought, well, this might just be true for these few children that have these rare mutations. But what's becoming clear now is that even in patients that don't have these mutations in TLR7, their disease is still driven by overactive TLR7. So finding some rare variants in some children have given us the clues to uncover what might be actually the central pathway, if not in all patients, in the majority of patients.
And I guess that means it could open doors of understanding for other autoimmune diseases as well, not just lupus.
Look, absolutely, because there's a few overlapping diseases, connective tissue diseases, that also have to do with, losing tolerance to some of our self-antigens, including self-nucleic acids. So we think this might also apply to a few of these related autoimmune disorders.
So now you know this information, where do we go from here? Is it looking into creating medicines?
Exactly.
Detecting for when we do blood tests, looking for certain components. Now we know this information. How do we make sense of it to prevent or protect and support those who may be?
So this is why it has been so exciting. So there are now clinical trials taking place as we speak, trialing small inhibitors of these molecules, specifically TLR7, the same receptor that is mutated in some children. And the results that are starting to come through now, there's a trial that's going to be published very recently, but the results have just been made public in a scientific meeting.
Okay.
So I can't disclose everything, but I will express my excitement that it does look as if, at least in all patients, inhibition of this molecule dampens a particular signature that we've associated with pathology, with disease, right? So very encouraged, very exciting. So it means that, perhaps in some time from now, patients will be able to benefit from this drug that specifically targets the key gene, right, in lupus. Many of these therapies might not be cures because once disease is established, right, unless you do something quite drastic. And there are there are some other alternative treatments that are, giving CAR T cells, for example, that destroy all of our B cells. So it causes a reset, right? There's very exciting, new trials coming from Germany where patients are, being depleted of all of their B cells. And then after that reset, they go into a deep remission. But then to prevent disease from coming back, some of these therapies also might be quite important. And in milder forms of disease, some of these inhibitors are already showing some clinical benefit. So even alone or in combination, we think some of these therapies might be effective.
I'd love to know how technology has helped to accelerate science research and how the technology used to support you in your studies has also changed throughout the years.
Yes. Technology has been key, right? When I started doing my postdoc, we didn't have a full draft sequence of the human genome. We didn't have the technologies to sequence whole genomes. So we had to, start with mice, do, large-scale mutagenesis of mice to find mutations that would cause disease and work back and try to see if these were relevant to humans. Now, thanks to technological developments like not just next generation sequencing, but then CRISPR-Cas9 editing, right? Genome editing, being able to very simply introduce a different base pair in the DNA. Not only we could discover these genetic mutations that cause disease in patients, but we could then introduce the mutations into mice, which, it's the only way to prove causation, to really show that that particular mutation in the patient, isolated from the rest of the genetic background of the individual, causes disease when you put them into a healthy mouse. And then we use mice also to understand how disease comes about. Of course, there are other technologies that we are using. There's all this explosion in omics, right? Transcriptomics, proteomics, metabolomics. And it's, being able to not only do all of these, but integrate all the data. And this is where AI comes about now, and all these, machine learning tools are starting to become not just useful, but necessary.
I'd love to, let's explore a little bit more about the data and AI side of things within genomics and immunology. I'd love to know how you're leveraging that or exploring its potential use cases.
We are exploring it because, first of all, the data, we get to a point that it's not manageable by hand, right? We are now sequencing not thousands, but hundreds of thousands of cells that are themselves very complex, many different subsets. Each of the cell can express at any one time thousands of different genes. And on the other hand, we're also sequencing the receptors. And, at any one time, we can express millions of different receptors in our B cells. So there's no way, any human person can make sense of that data unless you start looking for patterns and you have some informatic tools that help you, handle the data, right? So this is where AI is starting to become useful, right? But for that, you have to educate your programs with very large data sets. And we're still learning how to do all of that. Also for, tissue histology and to find, patterns of tissue pathology, AI is quite good, right? Once you've, fed large data sets and many images to one of these AI algorithms, they can themselves pick up patterns that sometimes the naked eye would not be able to pick up.
And for me, it sounds like as part of this research now, you're going to have multidisciplinary skill sets or interdisciplinary skill sets, knowledge of biology, knowledge of chemistry, knowledge of technology. So what are teams now being made up as part of this science research, other than just sitting in a lab, which is obviously very important, but it now seems now you need a wide team and wide skill set of people to help support you in that research.
Exactly. And first of all, of course, bioinformaticians are becoming key members of teams. I mean, I cannot do any of these complex analysis, right? And some of my PhD students are very courageous and they try and learn, right? But we do need experts that know how to handle these big data sets and do the more complex part of the analysis. But of course, right, all of our teams are now very interdisciplinary. We have, as you say, chemists and sometimes physicists that can do the complex image analysis, right? And we have, clinicians. We have, of course, biologists, and so it's actually quite exciting because everyone brings their perspective, and it's when everyone thinks together and brainstorms that these new ideas and new hypotheses arise. So for us, it's also a very exciting time in research.
I think one of the misconceptions that people may have with the sciences is that it's not collaborative. The chemists stick together, the physics people stick together, and the technologists. But from what you've shared now, it seems that like all loads of different people have a part to play in the conversation as well, which is very interesting.
Yes, and look, and that's where places like the Crick are quite unique and also help that environment, right? First, because the group leaders are really, being selected for being the best in whatever part of medical and biological sciences they work with. So here we have a community that is extremely diverse. It's exactly all of these backgrounds that we've just been talking about. And also the way the building is designed, right? You see all these collaboration spaces. So we are continuously bumping into each other. We have, regular chalk talks, right? Where people from different disciplines take you through their latest ideas. And there's a lot of cross-fertilization by us working so closely together, meeting regularly, and the whole building and scientific effort being so diverse.
Amazing. And I think that leads nicely onto my next question is, why is the UK a great place for research? More specifically, the Francis Crick Institute, because I know you've been around the world, right? China, Australia, America as well. So I'd love to know, what is it that the UK has with regards to research that has made you stay here and want to continue your studies and research?
I mean, look, traditionally the UK has been a wonderful incubator, of good science. Lots of Nobel Prize winners here, but the Crick is particularly special, not just because of its size and I mean, there's about 120 group leaders, diversity. We've just talked about interdisciplinarity. I mean, for me, sometimes to have access to technologies that are very far away from my field is what I've missed when I was working in smaller places. We also have incredible scientific technical platforms, right? That's with people that are specialists in their fields, very advanced imaging, advanced genomics, right? That makes it very attractive for researchers. But I think it's also the spirit and our motto is discovery without boundaries. And the way the place is set up, the spirit of the scientists working here is exactly that. Let's work together. Let's think big. And I have to say the funding model is also geared towards that because, I mean, we are fortunate, right? We do have very good funding, and funding enables scientists to take risks and to be bold and to think big. And and that obviously is important to come up with the great ideas and get to do things that you would otherwise not try.
Yeah, I think having a bit of risk and letting people explore their creativity could lead to some groundbreaking research. And I definitely get the energy and the spirit and the vibe here, because it's giving like an open door policy here, where, in other lab settings that I've been, it's usually like closed, the chemists are here, the physics lot are here, closed doors, closed labs. But here it's a very open space for collaboration, where I can be like, oh, what are you working on? Or what's that you're doing here? And I definitely feel that someone is just passing in the building.
Look, and it's not just collaboration. The fact that you're meeting your colleagues very regularly, I think it makes you also have the other more personal conversations, and we get together at the end of the day and just enjoy a bit of social time together. And that also makes it a really nice environment to work with, right? You are working with colleagues, but those colleagues end up being also friends. And I think there's a particularly nice environment here at the Crick for Scientists, right? I have fun coming here. I love my colleagues, right? And that makes it nice, right? There's not as much competition when people also get on and, learn to know each other a little bit more. So it is a nice place to work in.
And I guess you mentioned about working with people, right? We spoke earlier, and you sort of celebrating the talent that you're working with. I'd love to know more about the new generation of researchers and professors that are coming in and changing and leading the science research space. And how do you support them? How does the Francis Crick Institute support them?
First I have to say that we are very fortunate here because we do attract really exceptional researchers that come from all over the world. Also because they've heard about the UK, they've heard about the Crick, and we do have these very extraordinary people, right? I'm hoping that you can interview a few from my lab very shortly because they come from places that are from very far away. They've made big sacrifices. They're here because they're quite passionate about science, right? I think for me, it's probably the greatest satisfaction at this stage of my career, right? To see them becoming passionate and confident to the point where they come with their own ideas and they want to, think about their own hypotheses, and they get excited because they've tested their idea, not just my ideas. And that is, it's for me, it's my second family, right? You see them growing and getting to that point where they are starting to think about setting up their own labs, and it's very exciting for a group leader to see that happening.
It's good to see not only have they supported you in progressing in your career, but actually nurturing the next generation of talent who will one day maybe take over your place.
Yes, and I think it's some of the nicest part of our jobs to see that, right? To see them grow and become great scientists.